Greetings, everyone. Welcome to the part 5 of Basics of Cancer from Nurture Oncology. So today we are gonna talk about the role of immune system in the pathology of cancer. What role does immune system play when there's tumorigenesis? So immune system has dual roles. Initially, when tumor cells are introduced to the immune system, it tries to eliminate it via via tumor suppressive roles. So therefore, it has an anti-tumor effect in the beginning, which recognizes cells and kills it. But as soon as this process goes on for a long period of time, it leads to a pro-tumor effect via chronic inflammation, and this could be due to an infectious agent or without an infectious agent. Therefore, cancer is described as a wound that never heals because there is continuous chronic inflammation, which finally leads to immunoediting and evasion of the immune mechanisms. So, in patients who are immunodeficient, this is the reason why they are more susceptible to cancer because immunosurveillance is not available for them. And how immunosurveillance happens is the basic mechanism of immunology, that is, antigens and proteins, which are differentiated from self antigens, and neoantigens are presented by antigen presenting cells to the T cells, and then they are killed. This is the elimination phase of immunoediting. Immunoediting has two further phases. In the first phase, as we discussed, there are T cells which kill tumor cells, and if successful, there is no progression to cancer or neoplasia. In the second phase, there's an equilibrium phase where only some tumor cells are killed and some tumor cells are not killed, but they are also not metastatizing, or they are controlled and sustained by the immune system or T cells. And they're trying to adapt the T cells' nature. So, in this stage, the patients go in a state of dormancy. I mean, in terms of cancer progression, cancer cells go into dormancy. And then there's an escape phase where immunoevasion is achieved, and the cancer cells finally escape from the T cell killing and starts dividing rapidly, also progresses towards metastasis. Now, how do we know whether these phases happen? So, there were certain mice models where the researchers suppressed the cytokines released by T cells involved in killing of tumor cells or infectious agents, and they saw that there's rapid progression of cancer when those cytokines like interferon gamma were inhibited, which was a proof for the elimination, equilibrium, and the escape phases. The similar models were tested, and this theory or hypothesis was established. Another thing is there have been studies which show the correlation of tumor profiles with the prognosis of patients. Patients, so they see the cells which are infiltrating the tumor biopsies. If they're the cytotoxic cells, then those cancers or those patients are associated with a good prognosis because the T cells are killing the tumor cells and they are still in the phase of elimination. But when there's a increased amount of T regulatory cells, so they're associated with poor prognosis because if you remember, the T regulatory cells have an anti-inflammatory response. So it's basically suppressing the immune system and helping the cancer cells evade it. One potential area of research here is there have been certain studies which which see the pre-treatment and post-treatment biopsies and to see the effectiveness of immunotherapy or any form of cancer therapy, chemotherapy in solid tumors, especially glioblastoma multiforme and melanoma. Then one area which have been under boom is the gut microbiome composition. How does gut microbiome composition affect immune activity in cancer patient? This is a very famous research question. So if you're a postgraduate student, you can collect stool samples and analyze the microbiome diversity in patients across different cancer stages. and correlate these profiles with immune cell activity and cytokine levels. Then further research in each step of immunoediting and some research questions at the end have been discussed here. In the elimination phase, researchers are trying to restore the interferon gamma sensitivity because they uh, found out that this can increase the efficacy of checkpoint inhibitors like PD-1 inhibitor, PD-L1 inhibitors. These are the receptors present on the T cells and they promote suppression of these and killing of the T cells. So these checkpoint inhibitors, along with uh, interferon gamma promoted therapies, have been under progress, and trials of this are going on in melanoma and lung cancer. Then there have been also certain novel mRNA vaccines which are introducing neoantigens. These neoantigens will lead to an increased T cell response, and this have been under progress in advanced melanoma. Then there are also some studies which are seeing the NK cell mediated elimination and metastasis prevention. These NK cells target the circulating tumor cells in the blood and prevent metastasis. In the equilibrium phase, again, the researchers are trying to bring patients of equilibrium phase into the elimination phase by promoting interferon gamma signaling pathways on ovarian cancer patients and pancreatic cancer. Then CAR T cell therapy, which is successful in hematology patients, have been studied in solid tumors. I think these are phase 1 or phase 2 trials, especially in glioblastoma multiforme, pancreatic cancer, and renal cell cancer. Then in the escape phase, although we have lost most of the pathways, or there's little to do now because the immunoevasion has been successful by cancer cells. What researchers are trying to do is they alter the tumor microenvironment by changing the metabolic state or by inducing ischemia. So this been studied in triple negative breast cancer and non-small cell lung carcinoma because these are very aggressive tumors. And then we have certain trials on interferon-based immunotherapy resistance. That the here researchers are thinking how to overcome this resistance, and this is in particularly metastatic colorectal carcinomas. Certain research questions for cross-sectional studies or prospective studies, so retrospective cohort studies. So you can compare the immune cell populations and blood and tumor samples from patients at different stages of cancer and compare their profiles. You can also uh, see what immune-related genetic alterations are present in tumors from relapsed patients versus tumors from non-relapsed patients following immunotherapy. Then you can perform a retrospective review of immune markers, that is of example, lymphocyte infiltration, PD-L1 expression, and compare their outcomes, how long the median progression-free survival and overall survival of those patients. So you would have to analyze the pre-treatment biopsies and the available clinical data. Then this was the basic of inflammation and how tumor progresses. Now the question comes that why do certain infectious agents induce and promote carcinogenesis? Um, we have studied the basic before, but when an infectious agent is introduced in the body, the WCs act to eliminate it. And during that action, they release a lot of reactive oxygen species, nitrous oxides, TNF alpha, and other chemokines and cytokines. So these agents cause DNA damage and further cause mutations. Therefore, cancer, certain kind of cells which are under rapid division are more prone to DNA mutations. And This is what happens when an infectious agent is present in the body for a prolonged stage. You will notice that no acute infection causes cancer. You will not see a patient who had acute hepatitis B and progress towards cancer. It's always chronic hepatitis B going into hepatocellular carcinoma. So, because there's an increased cell turnover during chronic inflammation, cells are being killed, but there's increased cell turnover and DNA damage, mutations, and tumorigenesis. Further, once a tumor is formed, the chemokines which were released initially, they attract tumor-associated macrophages, which alter the cells present around that tumor. So that microenvironment leads to further increased proliferation and survival of tumor cells in that environment. Those chemokines lead to increased angiogenesis. Those chemokines leads to immunosuppression, release of IL-10, etc. Further, a key transcription factor have been studied a lot and reported into literature is nuclear factor kappa B. This in normal cells, this have been inhibited by inhibitor of kappa B. As you can see in this diagram, when a certain stimulus comes, it leads to degradation of this inhibitor of kappa B, which releases the nuclear factor kappa B. And this nuclear factor kappa B then migrates to the nucleus. This was found in the cytoplasm. Migrates to the nucleus and then causes transcription of its target genes, causing inhibition of apoptosis, increased inflammatory response, metastasis, and angiogenesis. So there are targeted therapies at each step of this nuclear factor kappa B cascade. And here's another diagram which shows the associated stimuli like bacteria, endotoxins, and KAGE protein due to H. pylori, then inflammatory agents, carcinogens, which lead to activation of nuclear factor kappa B. And then further downstream, because of activation, we get inhibition of apoptosis, inflammation, metastasis, etc. Therefore, as we know the background of infectious agents, how they work, uh, via research, we have established therapies which focus on eliminating those agents or increased immune response against those agents. Like we have antibiotics for eradication of H. pylori, which prevents gastric cancer. Then we have NSAIDs, which has an anti-inflammatory response. Then we also have the IKK inhibitors, which was the uh, which caused the destruction of the inhibitor of nuclear factor kappa B. Then we have vaccines for HPV and HPV vaccine, which causes prevention of cervical carcinoma and liver cancer. So here I would like to discuss some potential topics for students preparing for ICMR STS. HPV vaccine was launched recently in India, so you can do a study assessing the awareness of HPV vaccine. You can do a CAP survey. You can evaluate or see what factors affect girls to get vaccine to prevent their vaccination, or what held them back for not going for HPV vaccination. There are certain factors like financial constraints, because HPV vaccine is slightly costly. It costs around 2,000 to 4,000. Then, what are other cancers which HPV vaccine can prevent? So most people don't know that boys can also get HPV vaccination because it prevents laryngeal carcinoma, it prevents anal cancer. So you can also see the trend, or the you do a cross-sectional study validating the awareness among boys for HPV vaccination. Then you can do a retrospective study assessing or seeing the long-term side effects of HPV vaccination. You can stratify it in two groups. and age-wide in two groups and proceed with it. Another thing is, in this vaccine area, there was one very interesting aspect which is under phase 1 trial right now, that researchers are developing personalized vaccine, that is a one vaccine for every patient according to their tumor profile. These are mRNA based vaccines for advanced cancer. And if I would be a person in the audience seeing this video, I would have definitely searched this trials on this, because this is too catchy for me. Then there's another autogene cevumeran, BNT122, for pancreatic cancer. This is another vaccine which is under phase 2 trial with combination therapy, chemotherapy, and radiotherapy to suppress pancreatic cancer. Then here's a brief mention of certain infectious agents and their role as cancer-causing agents. You can narrow it down upon your interests and see the epidemiological profile in your country, which or your area, or what kind of infections are more prevalent, and see the type of cancer associated awareness of people regarding immunotherapy, awareness of people regarding vaccination. You can see whether the lower socioeconomic status people have access to HPV vaccination or other kind of vaccinations to prevent cancer. These are certain areas which you can work upon as a undergraduate student, because I receive a lot of messages for brainstorming sessions or for topics related to ICMR STS, and there are many enthusiastic students applying to it. I would definitely encourage you all to narrow your topics down using videos we upload on Nurture Oncology and get to a point where you can perform a cross-sectional study and submit to ICMR. So thank you everyone. I hope you enjoyed it. Put in comments if you want any other topics to be discussed, or if you have any doubts as such. Thank you so much.